Mute. There we go. Okay. Cool. Full screen. All right. Got it. All right. For realsies this time. This is the outline of the topics that we're going to go over. I'm going to give you a brief rundown with our eukaryotes, eukaryotes, and our viruses. Remind us of some of the basics with those guys. Then we're going to go over the genetic stuff. DNA replication, transcription, which is making the RNA, and then translation, which is making our protein. Then we have the metabolism stuff where we'll talk about enzymes and what they are and their parts. Remind you guys of redox reactions, and then we'll go over aerobic respiration. And finally, we'll touch on immunology. We'll kind of get in the basics of the innate response. We won't really talk too much about inflammation because we mostly want to get into this adaptive immune response and that stuff there. And we probably won't have time to go over this in more detail. We'll probably save talking about this in context for Thursday when we do the review for that. But what we made it through for everybody else is through number 4. So we'll try to do the same for you guys. All right. So to start off with, all of our microbes. All of our microbes, some of them are pathogens. And so a pathogen is basically a microbe that can cause disease. And that's usually from the infection process. And disease is any deviation from health. So we have those divided into 2 categories. Those that are made of cells and those that are acellular. And so what is a cell? A cell is a membrane-bound unit. That is capable of replicating itself. All right. And so when we think about our acellular versus our cellular guys, this will be viruses and prions. Viruses are not able to replicate themselves, right? They have to get into the cells and use the cell machinery in order to replicate. So they aren't actual cells. There are also a lot of viruses don't have a membrane around them. We'll talk a little bit about that, remind you guys about viruses here in a moment. Whereas prions are just misfolded proteins. And so they aren't actually replicating themselves or making copies of themselves. They're just causing normal proteins to misfold the way that they are misfolded. And so our cellular guys can basically be put into 2 major groups. These are going to be our eukaryotes, also known as prokaryotes. And our eukaryotes. Our eukaryotes consist of bacteria and archaea. Our eukaryotes, this will be our fungi, consisting of our molds and yeasts. Let me see how I was going to define this. Eukaryotes. Okay, so we're going to go over those in more detail in a little bit. This will also be our protista. And let me hold on real quick. Move these down. So I want to put more information on. Oh gosh. There we go. Really? Yeah. Anyways, our eukaryotes. Let me wall these guys off. Our fungi, our protista, and our helminths. And if we were to look at protista even closer, we're going to divide them up and go over them in a little more detail. But our protista includes our algae and our protozoa. And the helminths, these guys are our parasitic worms. So how do we define our eukaryotes versus our eukaryotes? Eukaryotes, they all have a true nucleus. And other membrane-bound organelles. Whereas our eukaryotes have no nucleus and no true organelles. We're going to talk about the organelles and what they are when we get into the eukaryotes in a moment, but for now, I want to focus on the bacteria and the eukaryotes. Another page in here for our eukaryotes. First and foremost, with our bacteria, yes, we talked about the archaea. The archaea are mostly our extremophiles. And we learned one of them that's associated with periodontitis. But our bacteria, we usually divide bacteria up into gram-positive or gram-negative for the most part, right? So our gram-positives, how are these defined? We have a thick cell wall, whereas our gram-negatives are defined by the thin cell wall and an outer membrane. On that outer membrane, we have a molecule called lipopolysaccharide, which we also know is the same thing as endotoxin. All right. Both of these guys also have an inner membrane that we call the cytoplasmic membrane. So if we were to draw these guys, our gram-positive cell might look like this in the inner membrane. The gram-negative might look like this with the inner membrane. And they've got DNA chromosomes, one big circle of a chromosome. It's not a very good drawing of it. Wow. I go all the way back. Okay. So it's got its DNA in a big old circle. And it's got ribosomes. And all of this is just hanging out in the cytoplasm. So here's its DNA in a big old circle, and then ribosomes. Again, this will be the DNA ladder, whatever. All right, and then our gram-positives have a thick cell wall. Make it real thick. Those are negatives. Thin wall. And outer membrane. And on the surface will be that LPS. Endotoxin. All right. What is the bacterial cell wall made up of? Do you guys remember? What traps the crystal violet in our gram stain? All right, so we've got Miranda saying peptidoglycan. Can we agree on that? Can. Peptidoglycan is what makes up that cell wall. Put this up over here so we can see. Okay. So this is the basics of our bacteria. No membrane-bound structures other than the main membrane as part of that whole structure. That whole structure of the inner membrane, the wall, and the outer membrane, if we have one, this whole structure is known as the cellular envelope. Moving on. Oh, you're good. Come back to it. Let's just sit on it for a second. Okay. This is recording, right? This better be recording. This is not recording. Let me make sure this is recording. Yes, it is. Okay. I just wanted to be sure because I didn't want all this to get lost and then it's not be there for you guys to study later on. Okay, so our eukaryote groups. Moving on to our eukaryotes. So our eukaryotes, we know they have a true nucleus. And then they also have membrane-bound organelles. Oh my gosh, Mom. No, I can't. Membrane. So these organelles will include the nucleus for sure. It's a membrane-bound structure. You're good. We're starting off strong. And then the nucleus. What does the nucleus do? It houses our genetic info. It's the control center of the cell. That's the nucleus. Then we have maybe the endoplasmic reticulum. But this is a little bit also known as the ER. It's a little bit more complicated than what we initially want to jump into. There's two different versions of it. The rough ER and the smooth. The rough ER. Why is it rough? Ribosomes. And they are for protein synthesis. And the smooth ER, what happens there is we are going to make non-protein macromolecules like carbohydrates or lipids. Then we have the Nycil-friendly Golgi apparatus. The Golgi apparatus is our like packing and shipping. And then we have the mitochondria. Yeah, the powerhouse of the cell. So what this really means is that we are making energy, right? And what kind of energy? ATP. Exactly. Good job. All right, so in the form of ATP, which we'll come to, of course, because we're going to get to aerobic respiration. So we're trying to bring this all kind of first full circle here. So everything that your cells need to do, whether it's making proteins, building new cells, copying the DNA, whatever, you need energy in order to drive all of that. And so first things first, in a lot of things, is making that energy. And that's going to be done in the case of our eukaryotes in the mitochondria. But what if you're not a eukaryote? What if you're one of these bacteria? Where do they make their energy? Where do they make their ATP? Does anybody know? They don't have mitochondria. They don't have any organelles. So with that, they're going to be making ATP or perform any of their respiration process, whether it's aerobic or anaerobic. In the cytoplasm and along the inner membrane. So we don't forget about that. All right, coming back down to our eukaryotes, continuing on with our organelles. Another one might be a chloroplast. This is for photosynthesis. We only find this in algae and plants. Notice that fungi are not on that list. So these are kind of our major organelles. There are other ones that can also be in the cell that have other functions. This is our basics here. So let's start in with our groups. The fungi. Those will be our yeasts and molds. These guys are capable of sexual and asexual reproduction. They're asexual structures that we need to know about. Conidiophores and sporangiophores. Conidio and sporangiophores or phores. Now, if we're talking about conidiospores, these are the actual spore-forming structures. Conidiophores are the type of molds that form those structures, but I'm not going to differentiate between the two on my test. So, conidio and sporangio. Conidio are the ones that are open. They look sort of like our penicillium, sort of like this. Right? And the sporangiospores, they will be covered. So they will look more like a dandelion, but with a case around it, maybe. Like that. I don't know what my coworkers are messaging me about. All right. Cool with the fungi. These guys are either going to be saprobes or what we would consider chemoheterotrophs. They can be opportunistic pathogens. And that. We're going to move on to the protista. This is going to be our algae and our protozoa. Algae, these are primary oxygen producers. Whereas our protozoa are basically defined by their motility types. The first motility type is going to be the ones that have flagella. This is going to be an example of these would be Giardia or Trichomonas, which we would have talked about in lecture today. The most common non-viral sexually transmitted infection. Next, wow, okay, that's just my moving on its own. Next would be the ones that are moving by cilia. We didn't really talk about any diseases that are caused by these guys. They exist, but we're not going to get tested on them, so we're moving on. Next are the ones that have pseudopods. Fake feet. These are also known as amoebas. This would include Naegleria fowleri, and acanthamoeba. And then we finally have the non-motile group. They're also known as apicomplexans. This would include malaria, as an example. All right, the next group after that will be the helminths. We have a whole other page coming up for. Our parasitic worms, also known as helminths. Our helminths are all multicellular creatures. They have organ systems and mouth parts and digestive tracts and things like that. They have complex life cycles. Yeah, cool. So I'm cool with that. Okay. And they all are categorized as animals. They fall into kingdom Animalia. So our helminths can be divided into two groups. The roundworms and the flatworms. The roundworms are called nematoda. Whereas the flatworms are called platyhelminthes. And then there's two different groups of flatworms. the tapeworms, and the flukes. The tapeworms are called cestota, and the flukes are called trematota. What else do I want to say about these guys? We can give some examples of them. Let me see, make sure I want to keep on track with what I've covered for everybody else so I don't miss anything for you guys. Here we are. Yes, let's give some examples for these guys. So our roundworms for a nematoda, an example would be what we just talked about in our quiz, which would be Ascaris lumbricoides, which is also known as the giant intestinal roundworm. But it also might include our pinworms, which are caused by Enterobius vermicularis. All right, some tapeworms. One of the tapeworms that we know and love. The typical tapeworm that we think of is Taenia solium. For our flukes, one of the ones that we talked about just a second ago, schistosomes, also known as blood flukes. And then we also might have the Chinese liver flukes. I'm not going to write them out, but that would be Opsithorchus sinensis and Clonorchus sinensis. All right. Yeah.
Says that's liver or river?
Liver. My handwriting isn't great. Okay. Oh, I'm trying to scroll it on this screen. I forget I have to do it on here. Okay. Viruses. Let's go over our basics for them first. First of all, viruses are obligate, intracellular pathogens, or even parasites. We can go that far because they are causing harm to the host that they are living inside. Parasite. They live inside and they have to live inside. They're going to use the host cell machinery to replicate. Viruses that infect bacteria are called bacteriophages. All right, so let's get into these guys. All viruses have two things. The first is going to be their genome. Always nucleic acid, but their genome can consist of different versions of this. And so it could be DNA, or it could be RNA. DNA could be single-stranded or double-stranded. Same thing with the RNA. And that single-stranded can be positive sense or negative. If it's positive, it could be read directly by a ribosome. So it's like our reading frame. The negative means we would have to make a copy of it in order to read it. So this gives us 6 different types of genomes that viruses can potentially have, whereas cells all have double-stranded DNA. If you're a cell, bacteria or eukaryote, fungi, animals, it doesn't matter that if you're a cell, your genome is going to be double-stranded DNA. All righty. Keep squeezing my pen really hard. So the other thing that all viruses have is a capsid. This is the protein coating surrounding the nucleic acid genome. And it is made of some subunits called capsomeres. Some viruses might have an envelope. In the case of a virus, if it has an envelope, this is a membrane stolen from the host. Host cell whenever it buds out. And not all viruses have that. Viruses without an envelope have a special name. They're called naked. This includes poliovirus. Poliovirus is just a capsid and a genome, and that's it. And so it's super, super tiny. And as it turns out, the ones that don't have an envelope are actually hardier. So our naked viruses are actually hardier and are able to survive better than the ones that have the envelope. So the enveloped ones, they are more susceptible to drying out. They don't survive as long, and they're in fact our weakest of all of the pathogens and the easiest to control in theory. But I want to point out, yes, polio being a very severe, disease to get associated with a naked virus, but COVID and flu and Ebola all have envelopes. So to put that in perspective, just because they have envelopes and they're weaker and easier to control, doesn't mean they don't cause serious disease. So don't judge a virus by its cover. So next thing, some viruses also carry extra proteins with them. Or enzymes or whatever. This would include HIV and its reverse transcriptase. All right. Some viruses will incorporate themselves into the host genome and become a provirus. In the case of bacteria that do that, this is the difference between the lytic versus the lysogenic cycle. In the lytic cycle, we are multiplying viruses, phages, and then they burst out of their bacterial host cell. This is called lysis. In the lysogenic cycle, the prophage genome inserts itself into the host bacterial genome. If the bacteria gets new genes this way that help with survival, basically causing disease. And this is called lysogenic conversion. All right, we ready? Okay, so Miranda is asking, is the temperate bacteriophage the same as a prophage genome? So this is a good clarifying question. Basically, temperate phages, sorry, that's my husband calling. Temperate phages are bacteriophages that can switch between lytic and lysogenic. And so when they are lysogenic, then their genome is inserted into the bacterial genome, and that's when it's called a prophage. That's whenever the actual term prophage is referring to the viral genome that has been inserted into the bacterial genome. So temperate phages are capable of switching between the lytic and the lysogenic phases. So I'll make a little note of that on here, since we had a question about it. Okay, next one. Jumping into genetics, y'all. Replication. Replication is making DNA copies. That's it. So essentially, we have first understand DNA is made up of nucleotides. DNA is deoxyribonucleic acid. It's made up of nucleotides that we abbreviate as A, T, C, and G. A is adenine. T is thymine. C is cytosine. And G is guanine. And A binds to T, and C binds to G. What is happening, dude? I don't know. I got some crazy coworkers. All right. Nucleotides. I was hoping they were talking about work, but no, talking about something else. I don't know, dirty dancing or something. I don't know what's going on. I feel a little left out, not gonna lie. All right, so get back on track. All right, so now let's talk, that's our nucleotides. And a nucleotide consists of a 5-carbon sugar plus a phosphate plus a nitrogenous base. And that nitrogenous base is what determines the A, T, C, or G. Alrighty, so DNA, when we're talking about double-stranded DNA, that makes up our genome. Our double-stranded DNA. This is going to be existing in a form that we call anti-parallel. This means that the sugar phosphate backbone, excuse me, is oriented in opposite directions for each strand. This is to say that if the first strand is 5' to 3', the other one is going the opposite way. DNA replication occurs in a method that we call semi-conservative. This is to say that the new daughter strands, 2 new strands, used one old strand as a template to create one new strand. So this would look like this. This is the original. And this would become like this. This. These are old. These are new. Semi-conservative. And to get into our all-time favorite part of DNA replication would occur in a fashion similar to this. I want to make it smaller than that. For this spot here where we're opening up is called the replication fork. This would be your DNA. And it's opening up. This would be 3, and this would be 5, and this would be 3, and this would be 5. Replication always happens in the 5' to 3' direction. And this means that when we're making our new strand here, we can start here at our 5' and continuously synthesize in, adding on the nucleotides from the 5' down to the 3' of that new strand. This one is made continuously. Anybody remember what this strand is called? The one that's made continuously? Starts with an L. Leading. The leading strand. Yes. I know it's been a while. Yes, yes. The other one is made in parts. It can only be made in the 5' to 3' direction also. So we make it in fragments. This one is called the lagging strand. Anybody remember what those fragments are called? Okazaki, there we go. Excellent. I don't know. So these fragments will be made in the 5' to 3' direction in fragments. And then we will seal those fragments together. Anybody remember the enzyme that seals them together? Close. So polymerase is the one that's actually making these enzymes. And so DNA polymerase is the one that's reading in on these guys. Ligase, excellent. So this purple guy here will be DNA polymerase. I know. Mine's not that great either. It's not my finest work. And my green here will be DNA ligase, which comes in to join the fragments on the lagging strand. So FYI, I know we've already made it this far into this, but I'm going to post all these online on Canvas. So I want you guys to make your own notes, right? Make your own notes that can go along with mine, but I will post all of this on Canvas too, so that you guys know. But, you know, of course, you know how big of a proponent I am of taking your own notes. I tried to force it on you, but yes, I will post all of this online as well. So that was our DNA copying. That is DNA to DNA. Now we're going to get into transcription, where we are going to make RNA copies from the DNA template. Now we'll talk about RNA. RNA is ribonucleic acid. It's usually going to be single-stranded, but the more important thing to remember about RNA is that it's also made of nucleotides. Its nucleotides are going to be A, U, C, G. A is adenine, C is cytosine, G is guanine. We'll write all this out again. And U is uracil. And so when we're going through transcription, we are basically using the DNA, one of the DNA strands, as a template that we will build our RNA strand off of. And so our first step in transcription is always going to be initiation. The enzyme that drives making our RNA from our DNA template is RNA polymerase. And it will start at a specific region on the DNA called the promoter region. And opens up and then starts in with its next step, which is the elongation step, where it's going to add RNA nucleotides that are complementary to the DNA template. Okay, well. Here is so bad. All right. So if your DNA was A, T, C, T, G, G, then my RNA would be U, A, G, A, C, C. And this will go on. Okay, come on. This will go on until we reach a terminator sequence. Doesn't matter what that sequence is or anything like that. Just gets to a terminator sequence, the RNA polymerase will fall off and the RNA product will be released. There are three major types of RNA that we need to know about: mRNA, rRNA, and tRNA. mRNA is messenger RNA. It's a working copy of our gene. It's an RNA working copy of the DNA gene. Ribosomal RNA. It forms the structure of the ribosome. Ribosome is the structure that is going to allow us to perform translation, which we'll talk about next. And then tRNA is the transfer RNA. It carries the amino acids that correspond to the mRNA codon sequence. All right, so let's get into the translation process. Translation also happens in just 3 steps. This is going to be the first one, which is initiation. The ribosome binds to the mRNA. And the start will be at AUG, which codes for methionine. It will look like this. Here's our ribosome. Here's our bottom of our ribosome. Here is the mRNA in the middle of it. And we get, I don't know why I made it that long, but I did. It is what it is. So this one might be AUG. And the next one might be something else. Who cares? And at these little 3 nucleotide RNA sequences is called a codon. This codon, AUG, will match up with the anticodon, which will be UAC. on something that looks like this, attached to an amino acid, methionine. This, let me get a highlighter. This structure right here, that's the anticodon. This whole thing here carrying amino acid, this is our tRNA. And our methionine is our amino acid that's going to bring in. So then the next tRNA that matches the next codon here, the next tRNA that matches that one would come in and bring in the next amino acid and would link onto it by a peptide bond, and that would be the elongation step. Amino acids corresponding to mRNA codons are linked by peptide bonds. And finally, we will get to the termination step. We get to a stop codon. These are nonsense codons because they don't code for any amino acid. They cause the ribosome to fall off and the polypeptide to be released. I have a lot more space under here, and I was like cramming it all in. All right, questions about translation? We good? So is it important to differentiate between eukaryote and akaryote gene expression other than the splicing? So it's a good question. With eukaryotes, to go back on that splicing, because I know you remember it, but other people might not. Eukaryotes have exons, which are coding regions, and introns, which are basically just garbage, non-coding. Exons have to be spliced together in the mRNA. Before translation can start. Good question. So the other difference that would occur would be that because our DNA is in our nucleus as eukaryotes. So eukaryotes, the DNA is in the nucleus. So transcription, basically making our RNA, this step that was up here, making our RNA has to happen in the nucleus. That's where our DNA is. And so then we can move the mRNA out of the nucleus and into the endoplasmic reticulum where our ribosomes are, and then start with the translation process. However, bacteria, because they don't have a nucleus, once they start transcribing their mRNA, they can immediately start translating it. They don't have to move it anywhere else because it all happens in the cytoplasm. So this lends to a process called co-transcriptional translation. And it is what the name is. I know it's a big word, but it is what the name is. Co-transcriptional translation. Meaning that we can start translation while we're still transcribing. All right, we ready to move on to chapter 10 stuff? Enzymes. Enzymes can basically, let me do that. Write this, right was bad. Enzymes could basically be in two categories. Simple, which just means that the polypeptide structure does its own work. It's only one protein. Or conjugated. It needs more than one part. Okay. So with conjugated enzymes, the sum of all the parts, the version that does all the work, the working whole unit, is known as the holoenzyme. You guys vaguely remember this stuff? Is it coming back at all? All right, holoenzyme. The protein portion of that is known as the apoenzyme. This is where the active site is that the substrate is going to bind on. Remember, the substrate is the thing that the enzyme acts on, does the work on. All right. The non-protein portion is known as the cofactor. Cofactor. Always. This can be inorganic, such as metallic ions. In which case it's just still called a cofactor. But if it's organic, like our vitamins, or like NAD, which we'll talk about in a minute, then this is called a coenzyme. Make sure I'm caught up with everything. Translation, enzymes, cofactor. Okay, regulating our enzymes next. So we're going to regulate our enzymes. This can be done either through competitive inhibition or non-competitive inhibition. Competitive inhibition. This happens whenever the inhibitor binds at the active site, competing with the substrate for that active site binding. That's why it's called competitive. In non-competitive, anybody know where the inhibitor binds then? Somewhere else, not at, right. So not at the active site. So Miranda's got it. This is going to be called the regulatory or the allosteric site. Not at the active site, so it's somewhere else. And by binding somewhere else, it changes the shape of the protein, and the protein can no longer bind at the active site. But it doesn't bind there directly, right? Then we talk about our different enzyme classes. Make sure I'm on the right track with this. Yes. There were 4 that I really needed you to remember. We don't have to go over all 6 of them, but 4 that were important. The first one I'm going to mention is going to be transferases. These guys move molecular groups from one molecule to another. move molecular groups from one molecule to another, transferases. The second one will be ligases. Just like DNA ligases, DNA ligase joins smaller particles together to make larger ones. All right, whenever we talk about the next one will be hydrolases. These guys break large molecules into smaller parts. And I hate to break it to you, but we did talk about these guys in the lab, right? So some of the hydrolases that we talked about in the lab were urease, gelatinase, and amylase, which broke down starch. So we talked about hydrolysis. Hydrolysis is an action performed by hydrolases. And so hydrolysis means that we are breaking apart larger molecules into its smaller parts. And we're also breaking water in that process. And so that's why it's called hydrolases or hydrolysis. We are lysing water or breaking water apart. And so basically we use the parts of water to seal off the ends of the smaller molecules that we're making. Essentially, that's how it works. And ligase is the opposite of that. And then finally, we had oxidoreductases. These are enzymes that are involved in electron transfer. Jesus, that was loud. That was definitely a lightning strike. I just don't know where it hit. I parked in the parking garage. It did. It was really loud. We're getting there. I want to get out of here too, guys. So we'll get there. Oxidoreductases are enzymes that are involved in electron transfer. So I'll move it up to the top so you can still see that bottom part there. So this gets us to that point of talking about redox reactions. So redox reactions are reactions where we have electron transfer. We're either giving up an electron to somebody or we're gaining an electron from somebody. So that's the whole point of redox reactions. Yes, okay. So this gets us into our major terms, oxidation and reduction. And which one is which? And so I'll remind you guys, do you guys remember my little mnemonics that I had for these? I had oil rig. You guys vaguely remember this? Oil rig, and Leo says grr. This is how I learned it was Leo says grr. Yeah. So oxidation is loss. Reduction is gain. Or lose electrons and you are oxidized. Gain electrons and you are reduced. And that is always what that word will always mean. We're talking about reducing. We're talking about nitrate reduction or sulfur reduction. We were talking about gaining electrons with that stuff. So oxidation, based on oil rig or Leo says grr, oxidation is loss of electrons and reduction is gain. What else did I say about this? Okay, and this is going to bring us into basically our next part, which is our aerobic respiration, which is also driven by something called oxidative phosphorylation. We are going to use electron transfer to drive phosphorylation, which means adding a phosphate group, an inorganic, what we used to call it P with a little I or something like that. It looks like pi to me. I don't like it, but inorganic phosphate, something. In this case, it's going to be ADP, adenosine diphosphate, and turning it into ATP, which is adenosine triphosphate, which is our energy molecule, which is the whole goal of cellular respiration. You guys ready for aerobic respiration again? Because we're there. Yes. All righty. So aerobic respiration has three major parts: glycolysis, Krebs, and the electron transport chain. There's an intermediate step in here, which we will talk about as well when we get to it. All right. So first step is glycolysis. The whole point of glycolysis is to take our 6-carbon glucose and turn it into 2 3-carbon pyruvates. This is the goal, is to make pyruvate. As silly as it sounds, it's what's going to be driving everything else going on later on. So the whole point is to make pyruvate. We also make 2 net ATP and some NADH. This guy is an electron carrier. We'll come back to this later when we come talk about fermentation. But for now, this is our step getting us to pyruvate. Our intermediate step, we take our 3-carbon pyruvate and turn it into a 2-carbon acetyl-CoA. Acetyl coenzyme A is what that is. And whenever we do that, we will break off a little bit of CO2 here. It's our first CO2 made in this whole thing. The whole point of that intermediate step is to make acetyl-CoA. Then we have the Krebs cycle. In the Krebs cycle, we're going to take acetyl-CoA and we're going to put it into this cycle. It's got several steps in it that you, thank goodness, don't have to know from my class. And then from that cycle, we're going to kick out some products. We're going to make 2 ATP. We're going to make some carbon dioxide. And we're going to make a whole bunch of our electron carriers, NADH and FADH2. These are the goal of the Krebs cycle. And then finally, we'll come to our electron transport. In the electron transport chain, you have a membrane, and this is either the folds of the mitochondria or it is on the inner membrane of bacteria. But either way, this is a phospholipid bilayer membrane. And on that membrane, you'll have these guys. Whatever, let's put 4 of them. Sounds good. It's wild out there, guys. And then we'll have this guy over here that we will call ATP synthase. This is phospholipid bilayer. ATP synthase. All righty. Anybody remember what these are called? These blobs that are in the middle? These proteins? This is in the electron transport chain. This whole thing is the electron transport chain. Anybody know what these proteins are that are going to get excited by the electrons? I'm going to help you out. It starts with a C. These are cytochromes. So yeah, it's okay, Miranda. So we're going to get to the proton motive force, no worries. These proteins here that are embedded in the membrane, these are called cytochromes. So this starts to sound a little familiar to you. Remember in lab with oxidase, what oxidase was testing for was cytochrome C oxidase. All right. That's this guy here. All right. So what happens is NADH and FADH2, whatever our electron carriers are that we made from above, these guys are going to give off some electrons. And they're also going to give off some H pluses. And as a result, they're going to revert back to whatever they were before. Whatever. Okay. And these electrons are going to go through all of our cytochromes. And as they do that, the protons get moved out. And so you'll have multiple protons from multiple NADHs and whatever. And so we'll get a whole collection. And these are all H pluses, just to be clear, but I'm just going to draw a bunch of pluses. This is what you were talking about, Miranda. This is called the proton motive force. This gradient that is created is called the proton motive force. And this is what is going to cause these protons to want to come back in because there's a whole bunch of protons on the outside. And so negatively compared inside, so it wants to equal out. So the protons come in. When this happens, we will also have a DEP being phosphorylated into ATP, which is the whole goal of this guy. Like this. In aerobic respiration in the electron transport chain, you will make 34 more ATP down here. So you can have a total of 38 ATP, as opposed to just the 2 that were up in glycolysis. Okay, but it doesn't end there. Of course not. So now you have these hydrogens that are building back up out here, and these electrons that are building up from the electron transport chain. So we can take 2 of the hydrogens and 2 of the electrons, and half of our O2 and that will let us make water as a byproduct. So if we're going through aerobic respiration, this is where we will need oxygen. It's the only time that you need oxygen in your whole body is for this. So this is why whenever you suffocate, you die within minutes. Because you need oxygen, yes, but why do you need oxygen? Because you need ATP. And so if I suffocate you and you die, it's because you ran out of ATP. And we can't make ATP if we can't sustain that gradient. And so the oxygen is getting rid of those protons so we can keep that gradient going for the proton motive force to drive ATP synthesis. So if you are a true anaerobe, an obligate anaerobe, can you still go through the electron transport chain? You can. You can go through it. You just won't use oxygen as your final electron acceptor. Okay, so anaerobes, true anaerobes, like that are actually able to do anaerobic respiration, can perform this using something else other than oxygen. So our anaerobes use something else here. I want to use a different color. Oh, geez, that was a surprise. I didn't expect that. Use a different color here. I can't really write it that small, but anaerobes use something else. Okay, anaerobes will use something else. And so some things that they might use here, nitrate. If oxygen is the last electron acceptor and it becomes H2O, it gained electrons. So it was reduced, right? GER. Leo says GER, gain electrons, reduced, right? What do we talk about nitrate doing in our nitrate broth? It goes from nitrate reduction. If nitrate was reduced, imagine using nitrate there instead of oxygen. That's nitrate reduction. That's where it would replace it. Remember whenever we were having sulfur reduction, making our black precipitates with SIM or with TSIA. Sulfur reduction is having a sulfate molecule there, and it makes H2S instead of H2O. That's what's really happening there. So you guys starting to see the bigger picture on how these are connected to what we were learning about a little bit? It's all connected, as much as we hate it. We can't compartmentalize it. This time it's actually related. We have some other kinds of bacteria that are different that are called facultative anaerobes. Facultative, guys, if they have oxygen, they will go through aerobic respiration. If there's no oxygen, they will ferment. Ferment means that they're going to stay in glycolysis. Whatever. All right. So if they stay in glycolysis, then they will make, I thought I had a reddish one, but whatever. They will make 2 net ATP here. And they will also have this NADH electron carrier left over. This has to be gotten rid of. They can't build up. They don't just disappear on their own. So they have to get rid of that electron carrier. In fermentation, they will feed that electron right back into pyruvate. And depending on what method they use to do that, to get rid of that electron, that either makes ethanol, lactic acid, carbon dioxide, or other products that we get from fermentation. Just feeding that electron back into pyruvate, converting it into other molecules as a result. They'll make 2 ATP instead of the 38 possible. You guys following? You tracking? Any of it coming back to your memory at all? A little bit? Okay. Yeah. It helps put it into perspective of what's actually going on with like bacteria and what we were testing for in that lab. Because now we're going to move on to immunology stuff. It's your favorite. What are you talking about? Unit 3, guys. All right, so immunology. In immunology, we have our white blood cells. What's the other word for white blood cells? Leukocytes. Excellent. It's okay. It's okay. It's going to all blur for a little bit. So we have 2 different kinds of leukocytes, the agranulocytes and the granulocytes. Literally just determined by staining. Our granulocytes, these will be, there's kind of 2 major groups of these. The fills. I really wish that I had switched these. Hold on, because I had it organized the literal exact opposite on my other class. And now my thinking is going to get weird if I have it switched just suddenly. Okay. Back to it. All right. Fills, these will be our neutrophils, our basophils. And what's the last one? Eosinophils, right? And then we also have mast cells over here. For our agranulocytes, we have 2 major groups for these. These are going to be our lymphocytes. There's 2 of these. Do you guys remember what these are going to be? T cells and B cells, exactly. T cells and B cells. And then we also have our monocytes when they're in the blood. But when these guys move into the tissue, they become macrophages, which I'm going to just write it like this. and dendritic cells. We'll also go ahead and create two subcategories relating some basic functions of these guys, because I want to jump right into the acquired immunity stuff, because that's the stuff that everybody sucked at, right? So we're going to get into that. But first, we'll have our phagocytes. The ones that are our major phagocytes are going to be our neutrophils. These are really important in our innate response for just eating up anything foreign based on their PAMPs on their surfaces, right? Pathogen-associated molecular patterns. These guys recognize the PAMPs, they eat them up and just digest it down, and then go and eat up anything foreign, any waste products, anything like that. Some other phagocytes that we had to learn about: monocytes, which then moved into tissues to form macrophages, and dendritic cells. We also had some important cells known as antigen-presenting cells. These will be monocytes and their derivatives, macrophages, the dendritic cells, and also B cells. So some key characteristics that I want to point out here: neutrophils are not antigen-presenting cells, and B cells are not phagocytes. Are we ready? Because we're jumping into MHC now. Because our antigen-presenting cells are going to be the ones presenting antigen and getting our acquired immune system jump-started. But how are they going to be presenting antigen, and how is that going to happen? So this gets us to MHC, which stands for major histocompatibility. Nope. Complex. MHC. Now, I am not talking about MHC1. I am not talking about MHC2. I'm talking about MHC in general. What does it do? So I know you two are the only ones sitting here, and Miranda's kind of engaging a little bit. Does anybody know what MHC does, what its basic function is overall? Anybody want to make a guess? I know this is where we get stuck, right? This is where we get stuck. The whole point of MHC is to present any antigen to T cells. It does not matter what kind of T cell or anything. MHC, all it's there to do is to be a handle to hold out antigen and show it to T cells. That's what MHC does. That's its major function in the immune system. It is just a way to present antigen to T cells, period. Now, having that in mind, now let's discuss MHC1 and 2, okay? So MHC1 versus MHC2. MHC1. What kind of antigen does it present? Inside antigen, also known as endogenous. MHC2 presents outside or exogenous antigen. Beautiful. All right, MHC1 is found on all nucleated cells. MHC2, which is almost every cell, MHC2 is found only on antigen presenting cells. Bring those guys back up just for a second so we can see them. Only those guys have MHC2. Only those guys can flag down your immune system and say, I found outside antigen. Only those guys. All right. Next, MHC1 is only recognized by CD8. MHC2 is only recognized by the molecule called CD4. So those molecules, CD8 is a molecule that recognizes MHC1. CD4 is a molecule that recognizes MHC2. For some reason, Siri heard me call out to her and started recording the whole conversation over there. All right, anyways. So we're getting there, yeah? So let me see how I want to approach this next part of it. What I did next, I basically stated that, and okay, I moved on to the next thing. So now we're going to move on to the T cells. So that's our MHC stuff. Now we're going to move on to our T cells. These are T lymphocytes. They start off their maturation in the bone marrow, continue their maturation in the thymus, right? So we have two types of T cells, T helper cells and cytotoxic T cells. Basically what happens, well, we're going to start out with a scenario. This is a macrophage, which is an antigen presenting cell. We've got some bacteria out here. These guys engulfed the bacteria through that process we call phagocytosis. This is outside stuff. Yeah, it engulfed through phagocytosis. And so if it is outside, it's going to present it on MHC2. Here's our antigen. Excellent. All right, beautiful. So the antigen, before we get too far ahead with ourselves, the antigen specifically is recognized by a marker called the T cell receptor. We already know, because we remember from up here on this slide, we already know that if it's being presented on MHC, the only cell that's going to respond to it is a T cell. So we already know, based on that, that this cell that's going to be responding to this has to be a T cell. It has to be. Only T cells respond to MHC. Okay? So this is a T cell, we know that for sure. If this is being presented on MHC2, what type of T cell is it? What recognizes MHC2 from up here? MHC2? No, so monocytes can be presenting it, right? But the thing that's going to recognize the MHC2 is going to be CD4. So the monocyte, the macrophage, the antigen presenting cell is going to be presenting the antigen on MHC2 because it came from outside. Because it's on an MHC molecule, we know it's presenting to a T cell. That's the only thing MHCs do. Because it's MHC2, we know that MHC2 is recognized by CD4. CD4 is only found on T helper cells. This process right here is going to cause activation through cytokine signals. That's then going to cause this helper T cell to proliferate and then differentiate. Always, we're going to get memory cells and regulator cells. We'll get some of these, but we'll also be getting some other very important effector T helper cells. Always, we will get both of these, Th1 and Th2. Both of these are going to still be CD4 positive. Let me move them a little bit. Taking space. They're still T helper cells. It's just now we're Th1 and we're Th2. Th1 regulates inside response, and Th2 is the captain that regulates outside stuff. We'll come back to it in a minute. All right. But now what happens, we can see with our T cells. Let's reframe a little bit. Over here, we have an infected lung cell, infected inside with something. Here's some viruses. So no, this is going to be MHC. We're presenting it to a T cell on MHC1. Put this antigen on MHC1. Because it's on an MHC, we still know that it's going to be a T cell that's responding. The T cell receptor sees the specific antigen. And we know this is going to be a T cell. But what molecule sees MHC1? You know, Carissa? Yeah, it's going to be CD8. Miranda got it up here, CD8. And our CD8 positive T cells are a specific kind of T cell known as the cytotoxic. But this guy can't do anything until he gets the signals from the Th1 to regulate the inside response. Then it can activate. Is this helping you guys at all with understanding MHC? I hope so. I tried to reframe it. So hopefully that has made it a little clearer. MHC is always presenting antigen to T cells. If it is MHC1, then it is presenting it to CD8. If it is MHC2, it was way darker than I expected it to be. So MHC2, it's expressing it to or presenting it to CD4. We have our colors going over here to tell us what's going on. So regardless of whether you are going to be having a viral inside infection or maybe a bacterial outside infection, your T helper cells that are your captains that drive the response for inside and outside, the T helper cells are CD4 positive. They are always CD4 positive, and they will always be activated by MHC2 presenting antigen, and MHC2 only presents outside. So you have to have outside to activate your captains that can then activate the inside response. All right, so let's get back down to our B cells, our last little stretch here. B cells. Our B lymphocytes, they mature only in the bone marrow. And basically what happens is you'll have a B cell. that'll have a receptor on its surface that looks like this. And the B cell receptor, does not have a blue or something? I don't know. I don't like my color choices here, guys. Let's do blue. This B cell receptor is known as IgD. So basically this guy, he sees outside stuff floating by in the lymph node, sees it on his IgD receptor on its surface, brings it all inside, and presents it on MHC because it came from outside. Then, because on MHC2, we know that the T cell receptor will see the antigen. What's the marker that sees MHC2? CD4, always. So we know this is a CD4 positive cell, and CD4 positive cells are T helpers. These guys will activate each other. Then these guys will proliferate and differentiate into TH2. It'll also make TH1, but we covered that stuff on the previous page, right? But TH2 is the guy right here. TH2 is the guy that activates basically the outside response. And so now B cells, as a result of that, oh no. These guys will proliferate and differentiate. We will make memory cells and regulator cells, and we will make effector cells that we call plasma cells. These guys make antibody. All right, so for our antibodies, first, we always make IG. You guys remember which one? So IgD is our receptor. It was up at the top up there. So first we will make IgM. Then we will go through something called class switching. And then we can make IgG or IgA or IgE. IgG is our workhorse, excuse me, workhorse. It's found in the blood. It's important for our secondary or amnesiatic memory response. IgA is our secretory because it is secreted in the mucosa. And then IgE is related to allergies or parasitic infections. Questions? Because this is going to be it for today. All right, so I will post this video on Canvas along with these notes in a PDF format for you guys. So you'll have these to reference. I know you made your own notes as well. So I'll go ahead and post these onto Canvas so you have this to refer to, and you can also use it to watch along with the video. Here's all my pages of notes that I made. If you made smaller writing, then you probably took less pages than I did. I'll post this. And then on Thursday, when we have our review on Thursday, we'll have our presentations first. I think we only have like 2, if I'm being honest. I'm pretty sure. It's like 2, maybe 3.
email,
Yeah.
And I was like, it first said, if you're getting this, then you have a report. I'm like, wait, what?
Yeah, you already did yours. I forgot to like actually assign it to the specific people. I just like made the thing. So I'm sorry about that. Yes.
The first thing I woke up to, like I woke up and I saw that, I was like,
Yeah, surprise. Yeah.
So confused.
Sorry. Yes. So, but yes, so we'll do our presentations on Thursday. And then after that, we'll try to have meta questions. And so the meta question that I'm approaching with everybody, so you can start thinking about it if you care, is going to be following through HIV infection. So we're going to have an HIV infection and we're going to talk about everything that's going to be going on in the cell, what HIV is, talk about transcription and translation in that respect. We're going to talk about the immune response and how the body's going to be reacting to it. We might even get into some discussion on like epidemiology sides of things and that sort of stuff. So we'll try to make it like literally just see where it leads us. And so on Thursday, everybody be prepared for discussing that stuff. That's going to be more effective if we have more participation with it, but I'll be here for those of you who are actually going to be involved in it. We do not have Bloom questions for Unit 4. I did not make them specifically for Unit 4 because most of Unit 4 reviewing for that stuff is really going to be basically, for sure, knowing whether something is a bacteria, virus, protozoa, or helminths type of a thing. And it's real basic kind of recall for that. But then understanding the concepts from Units 1 through 3 are going to help you answer the questions that are going to be relating back to the Unit 4 stuff. Because there's going to be questions that'll be like, basically asking like, which of the following, diseases is caused by a bacteria and that sort of stuff. So not just thinking about this is a disease caused by a bacteria, but like knowing, which of the following from the list are bacterial ones. What is the name of the organism that causes the primary cause of meningitis or something like that. So we don't have Bloom's questions for that. The only Bloom's questions that I have for those are the ones that are the open-ended questions on Canvas that we had posted on there. That's basically just like, write me down everything you know about the diseases. And then the questions for that stuff is going to be basically related back to our thinking from Units 1 through 3. So I don't have Bloom's based questions for those guys. Any other questions? Hey, I challenge you to write your own. Have ChatGPT write you some. And if you do, if you want to send it to me, I'll double check them for you. If you want me to. Any other questions from you guys? All right, then I'm going to call it a day. Thank you guys for hanging out. Of course, email me while you're studying. Thursday is our last day, and I'll see you on Thursday. Stay dry out there. All right, we're done. Get off of this. Stop. Boom. And